defense system of our country, then the T cells of our immune system is very much comparable to the armed military force. They are like soldiers, right? They are like trained soldiers. And in this video, I'll be talking about how T cells develop inside the thymus. But first thing first, before even thinking about what type of training do they get in thymus, we should first talk about how they are born and their birthplace. So the birthplace of T cell is the bone marrow, especially the big bones. Let's say the femur bone. So inside the bone, there would be specific regions. There would be vascular and endosteal niche. And in these specific niche, the T cell or the B cell is born from the progenitor, from the hematopoietic pluripotent stem cell. Now this hematopoietic pluripotent stem cell give rise to lymphoid progenitor or myeloid progenitor. And this lymphoid progenitor is the important one because this give rise to the adaptive immune system components, whereas the myeloid progenitor give rise to innate. Lymphoid progenitor in sequential step give rise to T cell precursor. T cell precursor are also progenitor by virtue, and also B cell precursor. Now B cell remains in the bone marrow and their development take place in the bone marrow, whereas the T cell precursor, they leave the bone marrow in order for them to go to a school which is thymus. So they enter the bloodstream and reach the thymus. Thymus is simply the training school of T cell. If we cut a cross section of the thymus, we get a view like that. Now thymus is anatomically subdivided into several regions, and we'll talk about that, its significance. So inside the thymus, there are thymocytes, which are immature T cells. There are also thymic epithelial cells. And the whole thymus is divided into several layers, like subcapsular region, which is the peripheral most region, then cortex, medullocortical junction, and then the medulla. The T cells enter the thymus from the high endothelial venule. Now, once they enter the thymus, they move towards the periphery and they gather in the subcapsular region. In the subcapsular region, these T cell progenitors can divide and amplify their number. So they really divide and amplify their number in the subcapsular region. From subcapsular region, they descend down to the cortex. And in this process, they encounter thymic epithelial cells, which give them important training. So thymic epithelial cells could be considered as the teachers of these T cells. At this point of time, these T cells are really mature. They would undergo several degrees of training and screening procedures throughout their school. From cortex, they move down to medulla, and they also receive another set of training. And after the medulla, when their training is done, they re-enter the high endothelial venue and migrate around the other parts of the body to the peripheral lymph organs, such as lymph nodes. It takes 3 weeks for this thymic training to complete. So it's a long time for them, right? And next couple of minutes, we'll discuss what type of training do they receive during these 3 weeks. So when the T cell progenitors are in the subcapsular region, they are known as the double negative T cell stage. It's a phase of their life. You can, just like we have our teenage, you can imagine the double negative stage is a phase of the T cell's life. At this point of time, they have the T cell receptor or the precursor of the T cell receptor, but they don't have the co-receptor CD8 or CD4. Now the T cell receptor could be either TCR alpha or beta, or other, it could be also gamma delta. Now among these double negative stage, there are substages, and these substages are characterized by specific marker expression. For example, in DN1 stage, c-KIT is pretty high, CD44 is present, whereas CD25 is not present. Whereas if you compare this stage to the DN4 stage, c-KIT and CD45 and CD25, all of these markers goes down. So based on this expression of these markers, you can kind of categorize them into specific phases. Now we'll talk more about these phases in a different video. But for simplicity, let's think about the double negative stage as a unified stage. Now, double negative stage is the time for the VDJ rearrangement of TCR genes. Just like the BCR genes, TCR genes also undergoes rearrangement, and that gives the diversity of the TCR and its ability to interact with so many different peptides. Now, first, the beta chain of the TCR is rearranged, and eventually and sequentially the alpha chain would be rearranged, but they don't get rearranged at the same time. Once both of the chains are rearranged, the mature TCR alpha and beta is formed. But there is also a possibility that it would form TCR gamma and delta. But it turns out that TCR alpha beta is the prevalent isoform over TCR gamma and delta. The reason I will be discussing it in a different video. But now, after this double negative stage, they descend down to the cortex. They are still in double negative stage. When they go to the cortex, they start expressing both the receptors. Now they are termed as double positive. They started expressing the CD4 co-receptor and the CD8 co-receptor. And already by this time, by the time of double positive stage or at the end of the double negative stage, their TCR gene rearrangement is done and they have a functional TCR by that time. Now, in this double positive stage, they have both the receptors. Now, in the cortex, they would like first interact with the thymic epithelial cells. Point to be noted that these double positive T cell stage is a prevalent stage present in the thymus. Almost 80% of the cells of the thymus are these DP double positive T cells. Now they would interact with the thymic epithelial cells. The double positive cells has to recognize the MHC bound peptides. So this is the first degree of their training and screening procedure. So their job is to understand peptides that are displayed on the MHC. In later point of their life, they have to do this job. So first, these thymic epithelial cells teach them how to recognize MHCs, be it a class 1 MHC or be it a class 2 MHC. Now, two distinct selection procedure takes place in the cortex. One we call the positive selection. In this selection, the teachers, that means the thymic epithelial cell or the thymus, ensures that these T cells who are double positive, they learn how to recognize MHC bound molecule. So they ensure the MHC restriction. And there is also a procedure of negative selection. In this procedure, they learn how to recognize the MHC molecules, but the high affinity interactions are kind of killed. So this particular negative selection ensures that cell tolerance is maintained and there is no autoreactive T cells generated. In case of many autoimmune diseases, this negative selection mechanism fails, which turns our own defense system against our body, which is detrimental. We will learn about these negative and positive selections slowly and in a lot more details. So first, let's say this particular T cell can recognize the MHC bound peptide. Now these peptides are mostly self-peptides produced by these thymic epithelial cells. In this particular stage, their goal is to get familiarized with the peptides associated with MHC. Now, if these T cells can recognize the MHC bound peptide, then they would proceed further and they would go for the next level of screening. And imagine if they don't or they fail to recognize these MHC bound peptides, they would die by neglect. And it turns out almost 90% of this population is dead because they cannot recognize MHC bound peptide with a decent affinity. So imagine how stringent this process is, right? Only 10% remains and they goes to the next level. The next level is in negative selection. So they had to learn how to select or how to identify MHC bound peptides. But they also make sure that you should not engage with a high affinity interaction. So the T cells which engage with the MHCs with a moderate level of affinity or a low level of affinity, they goes to the next level of selection procedure, or they proceed in their career. Otherwise, the T cells which recognize the MHC and with very high affinity interaction, they are dead by the negative selection procedure. They undergo apoptosis, and this is a method of generating tolerance. And at this stage, it is decided that these cells should not survive, because if they survive, they would turn against their own body, against our own body. They would create harm in our own body, and they can cause autoimmune disorders. So this thymic education is very stringent, and that makes sure the T cells gain specific skill sets, but they don't turn against our own body. Now the job in the cortex is done, and they have quite a lot of interaction with the thymic epithelial cells. Now they descend down from the cortex, from the subcorticomedullary junctions to the medulla, where they interact with other thymic cortical cells. And at this stage, the double positive cells either recognize class 2 MHC or they would recognize class 1 MHC. The subpopulation which would recognize class 2 MHC bound peptides, they would eventually stop expressing CD8 and upregulate the expression of CD4. Eventually, they would become the CD4 positive helper T cell. The population that would recognize the MHC class 1 bound peptide, they would downregulate expressing CD4, whereas they